Guten Morgen.
Good morning. We are talking about plant protection this morning, together with Falk Behrens from JKI. I'm going to talk about this topic. The first step is the analysis of the status quo. This was one of the first activities of the project when we started in 2019 and 2020. The objective of this status quo analysis was to find all the objectives, and we did a survey of all members in order to find out what their needs are, what they would like to see when it comes to plant protection and downy mildew. We carried out the survey in June 2019. Or rather, we started preparing it. It was then sent out to members in 2020. It was an online survey and via easy feedback. This was comprised of a mixture of open and closed questions. And we then carried out a quantitative and qualitative analysis of the responses. The questionnaire asked for information on the operations, about what the size, whether they did organic viticulture, etc. And then we started asking questions about plant protection and pewees to see how they work with pewees in contrast to traditional varieties. We prepared these questionnaires together with our experts and consultants who are in direct contact with practitioners. There is a report on the results of the survey, which is also available online in case you're interested in some more details. I'm just going to touch upon some of the results. Since it was an extensive survey, I can't cover it all. So 288 visitors, 121 took part in the survey, 96 completed it, and the rest seem to have stopped in the middle of the survey. But this is a good figure. We sent it to the members of all 4 associations and had raised the awareness of it beforehand. So here about the combinations of plant protection products. The blue bar is operations that have more than 50% pewees, and green ones operations that have fewer than or less than 50% pewee. And so it still looks like the axial blower is the most popular or most widely used machinery. We also asked about the frequency of crop protection measures and the volume of product, and whether this had been adapted to the individual plot. The responses were in general yes, and this shows how good practice works and how these wine growers treat their vineyards and see the differences and the different needs. And the wine growers that had more than 50% pewees adapted their frequency and volume more often than the others. And now looking at the average frequency of application of plant protection products. So here you can see that this is less than 5 applications, and the standard operations had an average of 10 applications. These are all organic wine growers, maybe we should stress that. And then we asked them about the amount or the volume of copper that they use for peewee varieties in the course of the years. And we should also say that after 2020, we had more fluctuations, actually, than you can see here. So we asked between 2015 and 2019. It's so interesting to see that the peewee wine growers had a relatively stable response over the years. So they hardly used any copper for the peewee vineyards. Now, looking at the traditional varieties, grape varieties, there is a more interesting result. You can see that between 2016 was a quite humid year, 2015 the opposite, and you can see that by the volume of copper used. But here we're always around 2 kilos, and only to 2016 the result was more than 3 kilos. And this shows that these organic wine growers usually remain around or below 2 kilos of copper, even for the traditional grape varieties. We also asked them whether they use other plant protection products that have not been maybe scientifically examined to such a great extent, but that they have found to be effective. And you can see the answers here in the word cloud. You can see that compost tea right at the center was big, and also effective microorganisms, and then also cadmium nettle tea. All of these are products that the wine growers use because they've observed a positive effect. And phosphonates were not used so frequently or mentioned so frequently as probiotics or microorganisms were. We then asked the wine growers where they see a need for research and development. And the answers were then divided into categories. First of all, about the diseases and the pathogens. Of course, here Pyrrha and Oidium were in the foreground. And you can see that here, when it was about copper and sulfur replacement, they wanted alternatives. They wanted to be able to reduce those substances. Well, this is no news. And then approval of active ingredients. Many wanted plant-based copper alternatives. Drones were mentioned for extremely steep vineyards. And when it comes to cultivation strategy and management, they talked about regenerative agriculture and also water and canopy management, as well as the effects of grafting on the health of the vines. And then when it comes to cultivation and varieties, they wanted more research into peewees, better selection, and more fungus-tolerant varieties. So anyway, alternatives to copper and alternative active ingredients were the most sought-after factors. And so this was the survey. This was right at the beginning of the project, and you can see it was interesting to examine the status quo. If we were to do this survey again after 5 or 6 years, it would be interesting to see whether we would get the same answer. The second work package within the project was the setting up of a practical research network. This practical research network was intended to include and integrate the wine growers' operations and to have a small group where wine growers were able to define their own success factors and contribute their own observations. This was limited to 10 wine growers, and then when we compiled the list of wine growers, we tried to spread them across regions, associations, and also structures of organization. And we then we published this via our channels to recruit interested wine growers. So we wanted to have some pioneers as part of the group, but also some who were not so experienced with research projects, but still interested. We then compiled the list of those wine growers. And then we invited them all together. At the beginning, it was online, and then we also had some face-to-face meetings where everybody was able to talk about their own problems and their own needs. Just something that they'd observed, and asking others to maybe try out the same things under different conditions. We carried out 3 experiments. This was without scientific support, I should stress. This was just an observational experiment. The first one was mowing measures. This was a wine grower from the Mosel region who had one steep vineyard where he had a lot of nettles growing. And he had observed that the vines were always healthy. And he wanted to know whether this was something that just applied to his plot or whether that would work for others. So he did not mow the nettles and allowed them to grow relatively high. And then there were a couple of other wine growers who also had steep vineyards and who decided to try the same thing, to not mow, and to see whether this would make their vineyards healthier or their vines healthier. The second experiment was about the reduction of copper to a maximum of 2 kilos in combination with potassium hydrogen carbonate. And we also considered reducing copper even more and to fertilize, to add leaf fertilizer. And the third experiment was the application of compost tea as a replacement, to start with, as a replacement of all crop protection. Mr. Behrens will say more about that a little later. And so we decided to do this as a replacement, not in combination with other crop protection products. We documented the seasons 2021 to 2025. And the 3 groups had 3 meetings a year by online meetings, once before the season and once after the season, to share what had happened, what had worked well, what hadn't. And in 2024, there was a physical face-to-face meeting which had a focus on compost tea and some training. So we had some expert presentations there. And some partners from the consortium were always present to be able to give us a little more scientific support. This practical research network was able to include someone from the consortium to research compost tea in more detail under proper scientific conditions. Compost tea is very popular, but there are a lot of questions, actually more questions than answers, and that is why this was a great way of linking a need that was voiced by the practitioners and to bring someone in from the scientific community to provide more information to the wine growers. So that was a very successful outcome of the project, in my opinion. So let me just present 2 seasons specifically, because there were some fluctuations between 20 and 24, always depending on the weather. And just to look back at the reduced mowing, this was great in the dry year. So this protected against the heat and had a positive effect on the quality of the grapes. It allowed the most moisture on the surface and in the upper layers of the soil to be preserved. Compost tea, well, we started analysis together with JKI and HGU, and we wanted to also analyze the build-up of agents, but we never, unfortunately, never got around to doing that. But we did do leaf analysis, and we also carried out some additional fertilization in order to provide enough nutrients to the vineyards. In 2023, things looked a bit different. The reduced mowing, this was very variable. There was more fungal infection in those vineyards where the weeds had been allowed to grow quite high. And this was probably due to the strong rain in August for over several days. And yeah, there was no erosion and washout because of the weeds. Compost tea saw the following results. There was no major problem with pyrofungus, and we also found that in those vineyards that had been treated with compost tea, there was less spotted wing Drosophila and other pests, but the vineyards that had been treated were stable for longer, but towards the end of the season, the resistance was not kept up, which is why we had a major loss ultimately. And then as far as copper is concerned, we reduced the use of copper in 2023, so we weren't able to draw any conclusions from that, really. I would now like to hand over to Falk Behrens, who's going to talk a little more about the compost tea experiments. Good morning. Good morning to everyone. I would like to talk about one aspect of the Practical Research Network, which was our experiments on compost tea, which and how it affects the microbiome of the soil surface. In 2022, the question was raised whether compost tea, which is defined differently by different people and which can be quite different in terms of its composition, whether we can actually see whether we can define its effectiveness. I was asked to join and do some experiments in order to answer two questions. First of all, how does the application of compost tea affect the microbial composition of the leaf surfaces, and which microorganisms are found on the leaves that have been treated with compost tea? Let me first define what compost tea is and how it's created. It is a filtered product of compost fermented in water, which means that we have here enrichments with useful microorganisms and extract from compost, which are applied either to the soil to take effect in the soil, or are applied directly on the plant in order to protect the plant surface against any infestation. And so here we looked at the application on the surface. First of all, this is you want to achieve an antagonism by certain microorganisms, and also systemic resistance to promote the systemic resistance of the plant. So this is how we went about it. This is the compost, made compost tea, which was then spread, and we then compared vineyards that had been treated and that hadn't. And we looked at all the, we used DNA extractions and used metabarcoding. So this is a typical microbiome testing to find out which microbiome we were able to define where. So we extracted the DNA from the samples, then multiplied certain areas, and then we were able to do a qualitative assessment, but also a relative quantification. The results allowed us to see how leaves treated with compost tea and leaves that hadn't been treated with compost tea differ in terms of the microbial consistency. And so you can see here, if the distance in this diagram is larger, the structure of the microbiome is larger, and if it's close together, there is hardly any difference. In addition, we will also assess the relative abundancy in order to find out which species were found on the leaves treated with compost tea, in order to see which one had been effective and which had established themselves on the grapes and on the leaves temporarily. We carried out this tests at Weingut Forster on the Nahe and at Winzer of Linde in Endingen am Kaiserstuhl. We carried out we did a first test in 2020 and then started taking samples in 2023 to 25. We had two plots on each of the vineyards, and this led us to 12 initial compost tea applications. And we then examined leaves and grapes over 3 weeks that had been treated and that had not been treated, in order to identify the microorganisms that had been established on the surface. Here are the results in 2023. This was one plot at Winzerhof Linde, where we found that the microbial structure of the leaves and the grapes did not show any major difference in terms of fungus. But when it comes to bacteria, we can see a shift of the microbiome. And we found that the treated leaves and grapes had 5% of microorganisms from the compost tea on them, which had become established here. And you can see here the first area are the untreated leaves, where sometimes you find sporadic microorganisms, but the microorganisms that we were able to attribute to compost tea were relatively stable and over time. These were mostly bacillus species, which are well known from biological preparations, and only one yeast in that year, which had been found in relevant quantities over the 3 weeks. 2023, as you may remember, was a dry year, and that, of course, made it difficult for the yeast. In 2022, it was a little different. Here, the yeast or the fungi played a major, bigger role. And now, here, this is 2023 at Weingut Forster, a similar picture, no differentiation in the fungus area, but a slight shift in bacteria on grapes and leaves. The share of compost tea bacteria on the leaves was a little lower, but it was still clearly existent. At Weingut Forster, we were also able to see that many bacillus were found, but also many other bacteria. And when it comes to fungi, there was not much to be found due to the dry weather in 2023. Now let's look at the conclusions from the first year. This method is very well suited to determine how the microbial effect of compost tea can be identified. And we also found that it's possible to establish potential antagonists by applying compost tea. And we wanted to be able to identify key microorganisms based on the 3 years of our experiment, taking into consideration their abilities to establish themselves in connection with the taking into consideration the weather. And this was to evaluate the application of compost tea at a microbial level. Thank you very much for your attention.